The topics include the Krebs or citric acid cycle, the electron transport chain, identifying the ATP synthase channel, chemiosmosis, and the substrate level and oxidative phosphorylation. First off, we have the Krebs or citric acid cycle, which is part of aerobic respiration. Its raw material is acetyl CO-A, which is coenzyme A combined with pyruvate from glycolysis. Each turn of the cycle produces 3 NADH, 1 FADH2, and 1 ATP. It occurs in a mitochondrial inner matrix. It produces ATP by substrate level phosphorylation, which will be detailed later in the video. CO2 is a waste product, and it turns twice for each molecule of glucose broken down. Next up, we have the electron transport chain. The ETC is a proton pump in the cristae membrane of mitochondria. Energy coupling reaction that creates proton gradient in the cristae membrane necessary for the production of ATP through chemiosmosis or oxidative phosphorylation. Energy released from the exergonic flow of electrons is used to pump protons across the cristae membrane to create a proton gradient. Electrons flow down the chain from one carrier molecule to the next in a series of redox reactions. The final hydrogen acceptor in the chain is oxygen. Water is a waste product and is exhaled. Next, we have to know about the ATP synthase channel, which is a protein complex that sits in the cristae membrane of a mitochondrion. It is responsible for producing ATP as protons flow down a gradient through the channel from the outer compartment to the inner matrix, a process called chemiosmosis or oxidative phosphorylation. There are hundreds of them in the cristae membrane. Next, we have chemiosmosis, which is where the most ATP is produced. It is the process by which ATP is formed in mitochondria as protons flow down a proton gradient through the ATP synthase channel in the cristae membrane. The cristae membrane is otherwise impermeable to protons. Next up, we have substrate level phosphorylation. This produces a small amount of ATP during glycolysis and the Krebs cycle. During this process, the enzyme kinase transfers a phosphate from a substrate directly to ADP, forming ATP. Lastly, we have oxidative phosphorylation. This is a broad term. It is the production of ATP using the energy that comes from the ETC to create a proton gradient and chemiosmosis. It produces the most ATP during cellular respiration.